So what we know so far there's some very good news, because people had to guess what the main antigen or the main thing to target on the SARS-CoV-2 virus was, and it was the spike protein. And in fact, that seems to be the right target. When we measure antibodies, neutralizing antibodies in patients that have had COVID-19 vaccine, we measure both their antibodies, but as well as their T cell response. It appears to be very targeted towards that spike protein. So that's good news for vaccine manufacturers. The COVID-19 vaccine appears to elicit very high neutralizing antibodies after a 2-dose schedule, somewhat better than SARS-CoV-2 natural infection, at least over the short period of time during the time that we've measured them. So following vaccination, we also have production of S protein specific memory T cells and B cells that are developed. And that was also documented in the clinical trials. So that's good news, is that, we measured not just one part of immunity, we measured antibodies, we measured T cells, we measured memory B cells. And so together with the high affinity SARS-CoV-2 antibodies, this hopefully will likely help prevent subsequent infection with SARS-CoV-2. So what we think it means, again, I'm putting my black cloud because that's what we think it means, is that, again, if we can elicit high enough neutralizing antibody titers, then maybe that'll be enough to kind of neutralize the virus, therefore blunt the size of the initial viral inoculum and avoid progression to severe disease. And hopefully, if we can prevent, that inoculum, there might still be symptomatic disease, but hopefully we can prevent, ideally symptomatic disease as well as infection and transmission. There's always going to be some virus that, get through the neutralizing antibody stage, but hopefully, again, with the fact that we're eliciting memory T cells, then those cells also can help and have the time to control the infection. So like I mentioned, there's definitely tons of studies, and also based on other pathogens or other infections, that show that neutralizing antibodies do wane over time, that re-exposure or a secondary, presence of the antigen will likely stimulate memory B cells to mount a rapid and effective humoral response. Humoral is, again, a kind of an antibody response. Decreasing antibody titers do not necessarily mean that there is waning or defective immunity. And antibody titers are expected to decrease following resolution of an infection. That's what it does. Again, I think that antibodies are a big part of the story, but not the whole story. And I think for certain population, and maybe even for the people with mild disease, the main protective effect might be their T cell immunity. We have some patients who from a genetic immune deficiency don't have any B cells, so they cannot make any antibodies whatsoever. So there's definitely been reports of patients with this kind of condition, so a condition where you don't make antibodies at all, that then acquire COVID-19, and they do okay, which means that they didn't have any antibodies and they were able to clear their infection. So likely it's a team approach. I think antibodies are important, but they're not the only part of immunity that you need for protection against SARS-CoV-2. There is increasing studies, different groups that are looking at, again, various aspects of immunological memory. This group has probably done the more extensive in terms of looking at both antibodies, but memory B cells and memory T cell CD4 and CD8 up to 8 months after infection. And I found that the majority of patients, so they studied a cohort of 188 patients who had COVID-19 infection and then showed aspects of immune memory in that cohort.